The third video on biochem. So I'm just breaking the slides up into 3 videos. All right, so this will be the third video, so let's pick up where we left off. So let's play from the current slide. So let me just review what we've done. So we talked about what biochemical molecules are. They're mostly organic molecules. Many of them exist as polymers. So they're typically really big molecules. So we talked about proteins. Proteins are polymers of amino acids. Proteins, of course, are a lot of enzymes, so they regulate chemical reactions. They help reactions happen. They are connective tissue, things like that, and they're long chains. Talked about carbohydrate. Carbohydrate is your body's first fuel. It's monosaccharides like glucose and fructose, polysaccharides like starch. Right? You should be able to recognize those. They're mostly involved in energy. And we talked about fats. Fats do not form polymers. They are non-polar. They're called lipids as a general term. There's 3 major kinds. There's fats, which are long chains, long-chain carboxylic acids. Typically, when they're stored, they form an ester with glycerol. So fats are your body's second choice for energy. After you run out of carbohydrate, you start burning fat. Right? Then the other thing about fats is steroids are a different type of fat. Steroids are, cholesterol is the parent compound. So cholesterol isn't bad, it's not good. Too much of anything is bad. So if you have too high cholesterol, that can be bad for you. Interestingly, your body makes cholesterol. So it was recently came out, there was all these dietary guidelines about cholesterol saying you should keep dietary cholesterol low, but it turns out that that's not really a big deal. Most of the problems with cholesterol are your body makes too much of it. In fact, most of the drugs that work on cholesterol, like Lipitor, prevent your body from making its own cholesterol. But cholesterol is the basis for bile salts, which help break down dietary fat so it can be absorbed. It emulsifies it. They're the basis for the steroid hormones. And then the last thing that fats are some of the other fats like leukotrienes and prostaglandins, eicosanoids are involved also in making hormones. And remember, hormones are signaling molecules. All right, the last major type of biomolecule are nucleic acids. These are DNA and RNA. All right, and this is information transfer. So the 2 major molecules, okay, they are polymers of nucleotides, so that's the monomer. DNA stands for deoxyribonucleic acid. RNA is ribonucleic acid. So the general idea is your DNA has all the information to make a new you. It's the actual blueprint to make you. Now, if you use the analogy of, let's say, blueprints to build like one of the buildings on campus. The buildings on campus there is a set of blueprints in an office somewhere in Sacramento that had to be approved, and that is the original copy. But when contractors are actually working with it, they have what's called a working copy, and that's going to have coffee stains and tears and things on it. It's not the original. RNA is the working copy. DNA does not make RNA. DNA has the information to make RNA. RNA does not make protein. It is the information to make protein. In this class, we're not going to go through how that works, how the information transfer works, and it's a time thing. We just don't have the time to do it. In the lab, spent more time on it, because we did a little bit more with it in the lab. So if you want to, for those of you in lecture that aren't taking lab, if you want, I can send you information. Just shoot me an email. Anyway, very, very interesting stuff. You learn about it in high school biology, and as I've said, it's fascinating. Very, very interesting how it works. All right, so these are what nucleotides are. So nucleotides are actually 3-part molecule. It's got what's called a nitrogenous base. Remember acids and bases? It's got a sugar and a phosphate. So phosphate you're familiar with. So the bases are called purines and pyrimidines, 2 different types. These are purines, adenine and guanine. Okay, you can see they're kind of a nitrogen base molecule. All right, a lot of nitrogen there. So adenine, and they're abbreviated A and G. Pyrimidines are these ones: thymine, cytosine, and uracil. Uracil is found only in RNA. The only difference between uracil and thymine is this CH3 group right here, if you look carefully. All right. And the way a nucleotide looks is like this. So this is the sugar. The sugar is called ribose. right? In DNA, there's not an oxygen right here, so it's called, or right here, excuse me, this is deoxyribose, that's from DNA, right? This is the base, sugar, base, and then phosphates. It's actually what's called a triphosphate, so adenosine triphosphate, ATP, you may have heard of. A lot of molecules biochemically have multiple roles. ATP is a big deal molecule, but we're not going to go into what it does. Okay, but these are what nucleotides are. Okay. Nucleic acid is a polymer of nucleotides. So if you back up, you can see what happens is two of the phosphates get taken off, and this hooks on to the sugar. Okay. And so you get this backbone. It's called a backbone sugar, phosphate, sugar, phosphate, sugar, phosphate, with the bases sticking up. Kind of similar to amino acids, where you have the nitrogen, carbon, carbon, nitrogen, carbon, carbon, and then the R groups hanging off. Right? Same idea, but that's how it polymerizes. Just a couple interesting things. Don't worry about sp2 hybridization. That's left over from an old slide. Bases are planar. They're flat. They are nonpolar. In nucleic acids, there's what's called base pairing. So there's a complementarity. Purines always, and the base pairing is hydrogen bonding. Purines always base pair with pyrimidines. Cytosine, hydrogen bonds with guanine. Thymine, hydrogen bonds with adenine. Actually, it should be thymine or uracil, hydrogen bonds with adenine. So DNA is double-stranded. So this is DNA. It's referred to as anti-parallel. So these numbers here, 5 prime, 3 prime, these are just kind of like head and tail, if you think of it that way. So there's a direction to this. See how it goes? Phosphate, carbon, sugar, phosphate, carbon, sugar, phosphate, carbon, sugar, as opposed to sugar, carbon, phosphate. See that? So there's a direction to this. So anti-parallel means it's parallel, but they're head to tail. So the head here is the tail here. But this is 2 strands of DNA. You can see if there's an A, there's a T. If there's a G, there's a C. If you were to build a big model, and that means if you peel this apart, if you separate this, if you know what the sequence is here, like the order of the letters, A, T, G, C, you can figure out what the other strand is. Right? If I know what this is, I can figure out this, because it's complementary. I just said that. It has a lot of implications in copying. And actually, one of the seminal papers in biochemistry was a letter to the editor of Nature. It came out in 1952, and it suggested how DNA might be copied. It's called a double helix, like a twisted ladder. So here's the sugar, phosphate, sugar, phosphate, sugar, phosphate. Here's the base pairing in the middle. It's kind of hard to see unless you know how to read these things. The blue ones are nitrogens. Think of it like a twisted ladder. Okay, and then I think the last thing we're talking about is that's one of the last slides. The way DNA probes work, and we spent more detail about it in the lab class again, is if you were to take DNA from a crime scene, and then you take DNA from a suspect or a random other person, since we know we can get the DNA, we know that DNA sticks to DNA, and the sequence is complementary, backing up, right? So let's say this is the crime scene DNA. I want to see, I make a copy of a suspect DNA, and I see if it will bind to this. Okay. So that's how DNA evidence works, so you can see if it's a match. DNA evidence is actually, fingerprints are more compelling because identical twins have identical DNA, but they don't have identical fingerprints. But everybody knows fingerprints are really hard to get. They can be smudged, right? They can be wiped clean if people wear gloves. DNA is extremely stable. So DNA evidence is, we always say DNA evidence today is kind of like fingerprint evidence was in the 1850s. In the 1850s, the general population did not understand fingerprints, how they worked. Now, of course, everybody does. The general population doesn't really understand how DNA evidence works. Maybe down the road in 20 years or so. Who knows? All right. Just some other things. Mutations are changes in the DNA sequence, and they can happen from the environment. So thymine dimers are caused from the environment, from sunlight, for example. It can have just damage. And DNA, when it copies, it's not perfect. It makes mistakes. Mutations are what drive changes in organisms. And there's a lot of detail we could go into if we had more time. But the way mutations work, it makes something a little bit different. Some mutations, the baby would never even be born. Some mutations will make the organism better. So mutations aren't bad. Mutations are the change. It's how that mutation interacts with the environment that determines if it's good or bad. And it just depends on where the mutation is. Imagine, if you had a sentence and you change one letter. It depends on what the letter is, right? If you could figure out the meaning of the sentence, it's kind of the same idea. All right, so that's the information on nucleic acid chemistry and the biochem. So again, what you want to be able to do is recognize this is a nucleic acid, this is a nucleotide, or that's an amino acid. Know what, just be able to, I'm not going to make you draw them, but I might do like a matching question would be really good for this if you look at the old exam. All right, understanding the roles of all of the biomolecules, which is really on the first slide. Understanding which ones are polymers and which ones are not, so you know what is a polymer of what. All right, and then just some general stuff about them, order things are burned for energy, that kind of thing. All right, so that's it for this information. I'll get homework up and posted eventually as well, and always available on Zoom. We're almost done.